welcome to the Westin MCAT podcast with your host, Ahmed
and Eric Olson.
Welcome back, everyone, to another episode of the Westin MCAT podcast. Today we've got Ahmed, myself, and Eric. We're going to be getting into a continuation of what we talked about a couple episodes ago when we started talking about replication. We'll kind of be continuing with the central dogma, and we'll define what that is shortly, getting into transcription today. But before we dive into that, Eric, any news on the Durham resident front?
Yeah, I'll tell you what's killing me right now is pathology. Or like histology. You'll have some classes in these first few months. So like, what is histology? I don't know. Did you do any histology in undergrad?
I had one histo course, and I actually quite liked it.
Go ahead. Yeah.
I was gonna say it took me a little while to know what I was looking for, as opposed to, those are a bunch of pretty circles.
Yes.
What are those circles?
What are those circles? Yeah.
I think it's one of those where if you catch yourself seeing patterns, you're in trouble. If you start looking at let me rephrase that. If you start looking at specific pictures over and over again,
yes,
right, you're in trouble. If you're just
it's not going to be the same every time.
It's literally never the same. Yeah. And I think for me, it was more so thinking about what structures would be present in a section of some tissue.
Of this tissue versus that tissue. And is it muscle or is it nerve and stuff like that?
That's actually, people might hear that and say, isn't it kind of obvious? And it's actually not super clear sometimes.
It is really tough.
For me, the hardest part was always the different parts of the GI tract. Do you have, glands under the whatever layer?
Yes.
It's a nightmare.
Well, so what's interesting about Durham path, and for anyone who doesn't, I think hopefully people are understanding what we're talking about, but pathology being, you resect some tissue or you do a biopsy or something, and then, you stain it with different stains and look at it under the microscope. And that's
exactly
ultimately how we make a lot of diagnoses. That's definitive diagnosis of a lot of things, is you have to look at it under the microscope and determine what exactly it is. And in Durham path, it's all skin. There's no are there glands, goblet cells or any of that stuff. It's all skin, so it shouldn't be that hard. But the dermatopathologists have decided to in dermatology, we say there are lumpers and there are splitters. And the lumpers are it's a cancer, whatever. It's some sort of cancer. It's a melanoma of some sort. Okay. And then there's splitters that are well, there's desmoplastic this, and there's, spindle cell that, and they will get into the box of melanoma and then divide it out into 50 different subtypes.
Oh, I see.
The way that our path lectures are scheduled, it's you do some reading on your own and then you show up and they put something up on the board and they're what is this? And you're I don't even really know what a lymphocyte looks like, white blood cell.
Yeah.
And so it's just a whole new language, derm, at least I had some context for, but you do very little pathology and histology in medical school. You do enough to kind of be introduced to it.
Yeah.
But I'm trying to get my feet under me in that. And it's something I am not good at. So it's a work in progress. But Ahmed, you started school this week, right? We're recording on a Tuesday, and yesterday was day one. So the people want to know what's
recording on a Monday, actually.
Today's Monday. Yeah, today's day one. There you go.
Yeah.
Days are running together for me. I don't know.
I was going to say, I'm pretty sure today was day one.
You're right. It's Monday, definitely.
Today was day one of orientation. It was a long day. Kind of as we were talking about sort of off air. It started with just general registration, getting my fob to access the medical building and different lounges and whatnot. Because our class is 280, they have to stagger it. And because my last name is AB, I was top of the list, which is not a good thing because that means 8 a.m. start. And the next event wasn't until 11, whereas some folks had 9:45, 10, even 10:15
Did
registration
you do some
times.
exploring and check out the areas? And...
So it's funny, and this is completely off the record, but over the summer, I was in Toronto, where I go to school now, and I was looking at different leasing, like properties that I was debating leasing, like just different apartments and stuff. And the I guess it was like the custodian or the janitor or somebody was like cleaning one of the doors, uh entering like the building, like from they were wiping it from the outside, and as they like opened it to enter, I just like kind of like slid my foot in to keep it open, and then just kind of walked in and toured the building. um
So
but
you're like ready to go already. You already,
yeah, and it's not a huge build. The inside is actually pretty straightforward. It was the outside that was kind of complicated because it's sandwiched within a lot of buildings. And so sometimes you lose perspective of like, oh wait, like this is still my building,
Mhm.
because it's just kind of surrounded by so much. That's especially important for me because I'm biking to campus. And my big thing was like, I want a bike rack that's like hidden from the streets, just
Mhm.
because like, nobody can just like be opportunistic and look and go, oh yeah, that's
It's quick.
worth my worth my effort.
Yeah.
And there are a couple of nice ones. But yeah, it's just the location of campus, like the medical building specifically, is really cool. It's like pretty much directly across the street from like the Ontario legislature, like the provincial
Yeah.
government and stuff.
Yeah.
And so you've got this big, beautiful government building across the street. And like there's a lot of scenery in the city. Like it's right on Lake Ontario, which is cool. But yeah, today anyway, like I was saying, registration, just getting like all my little like, uh, med student things like the fob and different necessities. And then um what did we do? We had like an academic orientation, and then we had like a big like class, like it was called a welcome picnic, but basically we just all gathered at a park and split up into groups and stuff like that.
At
Um
280 people, that's a lot of people to get to know, you know?
Yeah, so yeah, the groups were only like 20. So I didn't get to know everybody,
Yeah.
which and I actually don't know a ton of people in my class because like my school, my undergrad is like one of the least represented.
Mm.
I think in medicine in general, but especially at like the University of Toronto. And I think like just like there's two like big undergrad schools in the city that I'm from, and like from both schools combined, I think there's like seven of us out of the
So,
280. So like not a lot, and like of the other six, I only knew of one of them.
yeah.
Like
So
not
you're
even like
starting from
close
scratch.
friends.
Yeah.
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 But it's been cool. I'm relishing the opportunity to be like a new person again, because like my where I went to undergrad, I gone to high school in the same city and I'd been there for a while. And so it's refreshing when you know, people learn about you and they're like, oh, whoa, like I've like never heard that before, or that's so cool, or whatever. And like, whereas when I was, in undergrad, it was just kind of like because I had gone to high school and because, I had done a number of different things, like there was nothing really new anymore. Like
Yeah.
I just been around. But no, I'm excited for the week ahead. I've got already some learning to do when it comes to like actual school. We've got like these like self-learning things we've got to get done before the first lecture. We have a test on the first day,
Oh
which is
my, wait,
cool.
what's the first day? On Monday, next Monday?
Next Monday. It's
Yeah.
not a real test. It's just like it's like a demo of the testing software they use.
Hmm. Hmm.
Um, but yeah, it's frequent low stakes assessments is like the
Yeah.
model, which I actually kind of like
It's
because
nice. Yeah.
I need to stay on top of things and I don't have to study for something that's, super crazy heavy. But it's also pass fail, but it might be the highest cutoff for a pass fail I've ever seen. It's at 77, I think, is the pass,
Pretty
which is pretty
high.
high.
Pretty high.
But
You'll
yeah,
be
well,
fine. You're a smart guy. I'm excited to hear more about it as you kind of move forward and kind of hear how things are going and what you like
yeah.
and what you don't like and all of that.
The nice thing is the first 8 weeks are very undergrad like in the style of like information that's presented. So like it'll be a nice hopefully I'm not taking it for granted, but a nice kind of welcome to med school kind of period.
Yeah, and I will say, I mean, I don't know what all you'll be doing specifically, but I certainly found that like having tutored for the MCAT and like been, worked really hard to be good at MCAT knowledge sorts of things, like that
Yeah,
plays in your favor for the duration of medical school, but especially in those first couple of months as you go through like kind of the biochemistry and the genetics and the pharmacology and stuff like that.
yeah,
Like a lot of it is MCAT topics, and if you have a good
yeah.
in-depth understanding, which I'm sure are, more than ready for, then, it's like you'll learn some new things, um,
Yeah,
but you're in a good spot. You know what I mean?
yeah, I hope so. And I mean, we'll find out here soon enough. But yeah, I'm excited. Um, one of the like orientation week leaders like came up to me and basically said like, yeah, like you're not worried, like you're not like, because I'm like nonchalant to a fault. I'm like,
Mm,
hey, I'll figure it out. It'll be fine. And then just like you should have seen the look on like the three people that walked in earlier. Like there was there like you can tell there's a lot of nerves among everybody just
mm.
because it's a new everything. It's a new
Yeah.
life, it's a new style of learning, but like it's a blessing and a curse at the same time. The way I'm wired is like I don't get riled up really over anything. Like, I don't get too high, I don't get really too low. And like I don't like I usually don't get stressed. Like if like things are busy, they're busy, like chip away at it as best as I can and we'll figure it out. Um, doesn't always work out in my favor, might I add. Like it usually does, but not always. But I think the big thing for me was just like, Yerkes thoughts and law, right? Just like find that optimal level of like stress versus
Yeah.
casualness, I think would be the word. But in any
Yeah.
case, but yeah, no, it's exciting and looking forward to, what the next several weeks have in store. But in any case, as far as tonight's topic, the more important topic anyway, continuing what we talked about a couple weeks ago with the pathway, we'll say, from creating more copies of the DNA to pass on that genetic code to future generations of cells, and then using that DNA to, for now anyway, we'll just say do stuff. And I think let's start there with the DNA because the framework that we're kind of alluding to is typically accepted, from a naming standpoint as the central dogma. And
Yeah.
so, Eric, like, what does that actually mean? Like, what is the central dogma?
The central dogma is DNA to RNA to protein is really the long and short of it.
Yeah, that's
The
right.
way that I like to explain it to people is like the DNA is like a recipe book,
Okay.
and like the recipe book has all this information. Like when we talk about DNA, we talk about it as being like the the information to like make humans and make us function and stuff like that.
Right.
Um, and the things that are like making humans the way that we are and making any organism able to like function appropriately, are proteins. Like it's really all about proteins. And so you need the DNA, which is your recipe book, to make the protein, which is like effectively your, I don't know, whatever like baked good, I guess you want to do, like a cake, we could say. And the issue with the DNA is you can't just like take the DNA and like turn it into protein. There has to be some sort of process in place to like get from point A being DNA to point B being protein. And part of the issue that we run into in cell biology is that the DNA is located in the nucleus. And I think this is an important point to recognize when we talk about the central dogma and everything, is like locations of organelles matter for the MCAT. Like you need to know where things are and where they're functioning. And so the DNA is kind of sequestered in the nucleus, and it's like, a double phospholipid bilayer membrane thing that's like enclosing the DNA so that it's sequestered and guarded and safe away from everything else. But the problem is that we make proteins in ribosomes, which exist outside of the nucleus. And so we have
Right.
to somehow get our recipe for DNA, our recipe which is the DNA. We have to take that information and get it outside of the nucleus. And we do that by way of the RNA. So we take DNA and transcribe it into RNA. The RNA moves outside of the nucleus into the cytoplasm. And there it's used to make protein. So you could think about like, like if your grandma had a recipe book that was like, some old artifact and she had it for, decades, and she didn't want you to like mess with it, but you really wanted her recipe for, a gooey butter cake or whatever. I'm just making this up. I have gooey butter cake ice cream in my freezer right now, so it's kind of on my mind.
Man.
And
I haven't had dinner yet, so you're gonna have to.
oh, I'm sorry. Okay, we'll make this quick then. But the idea being like you can't take the DNA out of the nucleus, you just can't. And so instead you have to copy down that recipe. And that's really essentially what the RNA is doing, at least messenger RNA, which we'll touch later on different types of RNA. It might be for another episode, but we use the messenger RNA. It's kind of you writing down the recipe and then taking it to your house, to some other location, where you then do the mechanics of creating the protein or baking that cake. And that's really all the central dogma is again, what I said in the beginning, like it boils down to DNA to RNA to protein. I think those locations are important. I think the reason why is important to note. But that's really what it boils down to.
Yeah, I mean, I've never heard the cake metaphor before. For me, it was always like the DNA is the president, and the mRNA is like the military generals, and then the proteins are the soldiers, right?
Okay, I like that too. I like that. The president is like directing things and deciding what to do, and then the soldiers are carrying it out. Yeah.
Yeah, and you can't send the president out with the soldiers, otherwise you're compromising the state of the nation. And
I like that too. It's equally, makes the same amount of sense to me.
Yeah.
Whichever way is easier for you all folks to remember.
Yeah, whether it's cake or battle tactics.
Yeah.
Yeah.
You gotta pick one.
Right. Yeah. Two very similar ideas. But no, it's exactly what you said there, Eric. It's just the process of maintaining the DNA and then using it as a base to create these messengers that are giving these ribosomes instructions on making the proteins, which then go do just about everything.
Yeah.
Yeah, I think, it's important to note some of the structural differences that occur in the process of making that mRNA. Like there's certain things that the MCAT does like to test with respect to DNA and RNA. And sometimes like just being mindful of those differences can really help from a process elimination standpoint. Like sometimes, especially when you're given structures or sequences of of DNA or of RNA, the MCAT will sometimes throw in sequences or structures of the other, type of nucleic acid, and just being aware of those differences is really important. And I think the two major ones are three major ones. The secondary structure comes to mind, right? The DNA is double-stranded, forming a helix. The RNA is, in the vast majority of testable scenarios on the MCAT, single-stranded. Can have some secondary structure, like it can form little complementary bits within its strand, but still single-stranded.
Yeah.
The nucleotides, the thymine from DNA gets replaced with the uracil, and then the sugar within the nucleotide is a ribose as opposed to a deoxyribose, and that is just specific to carbon number 2, the presence of an OH as opposed to just a hydrogen, the OH being present in the ribose. And on that note, just if folks aren't familiar, a ribose or a deoxyribose, it's a 5-carbon sugar that connects, a phosphate to the nitrogenous base in a nucleotide. That's why the pentose phosphate pathway is very important in making nucleotides, right? It makes pentoses, 5-carbon sugars. And so just working around that sugar, you've got your nitrogenous base on carbon 1, carbon 2, with the exception of just differentiating DNA versus RNA and all, in the grand scheme of things, not super important. Carbon 3 is where we have the OH that links to the phosphate group on carbon 5. And then again, carbon 4, in the grand scheme of things, fairly irrelevant. Any like tips or tricks to remember those differences between the DNA and the RNA?
No, yeah, I mean the T to U is pretty. I think people probably, hopefully, are familiar with that. Like that's one that you definitely need to know. And this is exactly the case that Ahmed mentioned. Like they'll ask you for, the complementary strand to, or like they'll give you a coding strand or something, and then they'll say, "What's the RNA sequence for this strand?" And they'll give you one. They'll give you two that are exactly the same, except one has T's and one has U's, and you have to say, "Okay, well, we're talking about RNA, then it's going to be uracil."
Right.
The other thing that like it's not really a tip to remember these things, but I guess you could maybe view it that way. I think if thinking about like overarching implications of the structural differences, like the nitrogenous base TDU, like I don't know, there's probably something that some biochemist knows somewhere about like how the reactivity is different or something. I don't know what it is. But to me, the other two speak to kind of the stability of DNA versus RNA, right? Like
Yeah.
RNA has an extra hydroxyl group, and that's like a reactive species. Like that's a nucleophile. It has electrons that can be used to react with things. And so it's like more vulnerable to, chemical reactions in whatever sense. And then similarly, like single-stranded versus double-stranded, when you think about DNA, it's double-stranded, like all of the pieces that could be reactionary are like kind of on the interior of that DNA molecule. And then you have this like very negative backbone, which is basically like repelling everything from messing with it. And so DNA is like super stable. And I'm sure you guys have heard like, we can isolate DNA from samples that are like thousands of years old because it just doesn't degrade. Like it will just sit there
Right.
forever. It's a super stable molecule, whereas RNA, it has that extra hydroxyl and it has just the one strand, right? It doesn't have a complementary strand to like create a more stable structure. And so it's more um yeah, more unstable. Like things will react with it, things will degrade it. And we'll get into that a little bit later, is like when you send it out into the cytoplasm to get translated into protein, you have to modify it in certain fashions so that it will have greater stability because otherwise it will be degraded, just because it's a
Right.
less stable molecule than DNA.
Yeah, and I think it's important to understand I think the why of, why RNA is degraded, so quickly. And I think sometimes we have a predisposition to say, oh, like if something is degraded, that's a bad thing. But in the RNA case, it's actually a great thing because we don't want to overexpress certain proteins. Like, for example, there are very, aggressive enzymes that can be transcribed, and we want to be able to regulate those enzymes or regulate, certain membrane receptors so that we're not overdoing any type of, physiological response. And so the intrinsic sort of time clock or the lifespan of the RNA being what it is important because it's always easy to just make more RNA. Like that's a process that can be done, you know, fairly seamlessly, but not being able to degrade the RNA is an issue because now we get overexpression, which can lead to, well, overexpression in general, depending on what it is that's being overexpressed, can lead to cancer. It can lead to different, severe phenotypic abnormalities. And so we always want to keep things within, a homeostatic window. And so having that RNA degradation is important. And importantly too, like there's different ways of just regulating the protein products that they lead to. For example, enzymes could be activated or inactivated on demand as opposed to just needing to continue to make more enzymes, if that makes
Yeah.
sense. And so
Yep.
there's the just uh, the safety. Safety is not the right word, but there's this like, yeah, I guess like cell safety, I guess, if that makes sense. But there's also like the economic aspect of just like energy and not like wasting it, that comes with just like regulating the protein outcomes. Um, but getting into, the actual like process of transcription. In essence, what we've got to do is we've got to take that double helix. open it to expose those nitrogenous bases, and then we've got to read them and write them down essentially. And
Yeah.
that's what the verb transcribing means, right? To transcribe something is to take something and put it into text, right? Or to put it in writing. And so you can think of, your RNA polymerase as, the pen that's writing down the sequence, right? And then that sequence can be translated, but that's a story for another day. Um, where do we start here with transcription? Like, obviously, we have to expose the helix. What does that look like? Like, how does that process get started?
Yeah, I mean, it looks similar to what we talked about, I guess, 2 weeks ago now with DNA replication, right?
Right.
Like you have a similar kind of cellular architecture that comes in there and will open up the helix and allow you to create a new strand via RNA polymerase. But kind of on the subject, Ahmed, as you're talking about like this tight regulation of products and RNA and proteins and stuff like that. I wonder if you could tell us a little bit about like regulation of transcription, like promoters and enhancers and stuff like that.
Yeah.
How do those come into play when we talk about what's getting transcribed and what's not?
Yeah. So the promoter, let's start there. The promoter is the DNA sequence that the RNA polymerase has to recognize, and then it can start proceeding with the transcription.
Mm,
And so it's basically it's the waving flag sort of to tell you, hey, like I'm here, like start here.
yeah.
And what an enhancer is, it is a sequence that can be upstream or downstream, and it's usually pretty far away from the actual gene, where transcriptional activators can bind, and then it basically causes the DNA to fold towards the promoter. And then now we've created this transcriptional complex that can facilitate transcription. Um, What's interesting to note is that with eukaryotic cells, we're only transcribing one gene at a time. It's what we call monocistronic. Whereas with prokaryotes, they have this operon structure where multiple genes will share the same promoter, which is not the case in eukaryotes. And so that's why we get polycistronic mRNAs with prokaryotes. That is
Right, where
testable.
where you're transcribing multiple genes at the same time, kind of in one long strand.
Exactly. Exactly. You can almost equate it to writing down notes for multiple, classes on the same piece of paper. Okay. And there's functional differences there, and that's probably a discussion more so for our translation episode coming up at a later date. But in terms of the regulation, really it comes down to, do we have the transcriptional activators binding to their enhancer sites to recruit RNA polymerase to the promoter? Once
Yeah.
we've brought RNA polymerase to the promoter, these other enzymes can now start to go to work. Or the enzyme and these other activators and cofactors can start to go to work, where we can open up the helix now. And unlike replication, we only need to go in one direction. We don't need to go both ways. And that actually has to do with the nature of the strands. And so what I mean by that is we've got two sets of names for our two strands. We can have the template and coding strand. We can have the sense and I think anti-sense strands.
Yeah.
And so template versus coding always made the most sense to me. Template
Yeah, this
is
this is a I'll just say this is a tricky thing, and this is important to pay attention to because this comes up too. Like they'll put these terms on the test, like you obviously know what this is, and so if you don't, like you're up the creek, you know. So
yeah,
this is important to pay attention to and really get down, because these terms will certainly come up at some point.
definitely. The template strand for me made a lot of sense. It's the template. It's what you're creating that RNA off of.
Hmm.
The coding strand is what the code will look like, right? Because if you think about it, the coding strand and the mRNA are both complementary to the template strand. The difference is just there's uracils in the mRNA, whereas there's thymines in the coding strand.
So the coding strand is essentially equivalent to the newly formed mRNA. So basically what you're
Exactly.
saying is, if I can, sorry to interrupt, but
No, go ahead.
so you have a coding strand and a template strand, and those two are bound. Those are like
Correct.
complementary strands of DNA. And
Yes.
effectively what you do is you open up the DNA strands and you attach all your cellular machinery to the template strand. And you're
Correct.
reading off of that template strand and creating a complementary mRNA to the template strand. So it's going to be like the opposite bases from the template strand. But because that mRNA is binding to a template strand and the coding strand had also bound to the template strand, and that is, like you implicated, the coding strand and the mRNA that we're newly creating are going to be the exact same. So again, like I'm sorry to belabor this point, but like I've just seen a lot of questions like this where
No,
they'll give you
it's
one of
true.
these, they'll give you one of these, they'll say like, here's the coding strand and what does the mRNA look like?
Right.
And like one of them will be exactly the same as the coding strand, complete with thiamines. And you're like, okay, well that's not the case because an mRNA would have uracil and not thiamine, right? And one
Right.
would be complementary to the coding strand, which we're calling the template strand, right? And that's not going to be the mRNA because the coding strand and the mRNA should have the same sequence. So just this kind of thing. And like
Yep.
you'll have four different options and there'll be these DNA sequences or RNA sequences, and you have to be able to know what's the coding strand, how does it relate to the mRNA?
Yeah.
And that, that's a question that you're either going to get right or you're going to get wrong, depending on how comfortable you are with this terminology. So I think I,
Yeah.
it's not on every question, right? But like,
It
it's
comes up.
it will come up, and if you know it, then you get a question right, and that means something for your score. So
Definitely.
I think it's important to know.
I agree. I wholeheartedly agree. And the saying, the enemy of my enemy is my friend kind of
Yeah.
fits in nicely here. I don't know what's with all the war references today. Today was a
Yeah.
long day. Maybe
Long
that
day from it.
long peaceful day. Let's put that out there. But yeah, no, like if you think about it, like the point being like, two entities have a similar thing in common, right? So if I am the opposite of this, if A is the opposite of B and C is the opposite of B, then A and C must be similar,
The and C are similar. Absolutely.
right? And so that's kind of the idea here between the coding strand and the mRNA is they're both complementary to the template strand, and thus that makes them similar.
And so
As far
how do the sense and the anti-sense,
exactly,
how does that fit?
sense and anti-sense are just other ways of saying those same things. It's less commonly used. It's like the vast majority of the time um it'll be coding and template. If I remember correctly, the sense strand, the way that I remembered it is like the sense strand makes sense. It's the code. The
Yeah,
anti-sense is the one that I guess doesn't make as much sense with respect to the code because it's the template which is complementary to the code. And
I think you're
so
right. Yeah.
the thing that gets transcribed, so the strand that is read is going to be your antisense strand.
Yeah.
And
Just
the
like the
one
templates. Yeah.
exactly the one that is more or less identical to the code, just with that nucleotide substitution, that's going to be your sense strand. And
Yeah.
so, fortunately, these names make sense. Like it's conceptualizable. That's not a word, but we'll pretend it is. Um, the sense strand makes sense because it resembles the code, and the coding strand is the sense strand. I think it's not too big of a leap to make those connections there. And I think if you do, it just becomes a lot easier to keep those in line. Like I said, more frequently it'll be coding and templates,
Yep.
but I have seen sense and
Good to know.
anti-sense
Yep.
on the MCAT, so it's important to be mindful of both. Um, and
I always
so
remembered sense and code are both one syllable. And I know it's like coding, right? But like you can
yeah,
make coding into one syllable with code, but you can't make template into one syllable. Like temp
yeah,
doesn't make sense. And
yeah,
so anti-sense and template, like they're both two syllables, and
yeah,
code sense are both one syllable. So like those are equivalent terms, right? The coding
right.
strand. The
I've
antisense
never heard
emergency
that before.
transcription. I mean,
That's
that's
really
like
good.
so basic, like stupid mnemonic, but
I
it
like
works.
it. Yeah, no, I think it's
I
great.
will say, we want to touch on post-transcriptional processing before we wrap up, but I will just say something we didn't really touch on, and I was thinking about it, Ahmed, when you were talking about promoters and enhancers and things, like there are sequences of DNA in the, genome, which is basically like our term for all the DNA that we have. There are sequences of DNA that don't code for anything. And so like these promoters and
Yeah.
enhancers and repressors and things like that, those are all like non-coding DNA.
Yeah. And I can't
And
remember the exact number, but like more than half of the, like way more than half of the DNA is non-coding.
I think it's like I was trying to remember too while we were talking. I'm pretty sure it's like 99% is non-code.
Ye-
It's like a huge amount is non-coding.
Yeah.
And so
And
like go ahead.
I was just gonna say again, it's important to understand like why that's the case. And the reason being now, if there's an inevitable mutation, there's a huge likelihood that it's going to do nothing,
Yeah,
which is great.
it's true. It's interesting. Like we don't know all of what all of that DNA is doing, and like I think for the purpose of the MCAT, it's just important to know that there are parts of DNA that are not transcribed, and even within, as we get into this discussion on post-transcriptional processing, even within like the larger messenger RNA that we have transcribed, there are parts that will get spliced out, right? And those are termed introns versus exons being like the parts of the RNA that we're going to keep that are like the real code that are actually going to be translated in the ribosome into a protein.
Right.
Um, the introns are like specific to a gene. And we probably should have spent a little bit more time. I don't know if we have time today to really talk about like the differences between a gene and an exon and an intron, all those things. But
Mhm.
like the gene really refers to like all of the DNA code that contributes to the formation of this RNA. So like an enhancer is not getting transcribed, a promoter is not getting
Right.
transcribed, but it's part of the gene for that protein. It's part of like the DNA structure that will make up this protein. Like it's participating in the transcriptional process, even though it's not actively being transcribed.
Right.
So the gene will include, these enhancers, it will include promoters, it will include, all of the exons that end up like remaining in the messenger RNA molecule. It will include the introns that get spliced out. And once we get our initial messenger RNA transcribed, then, as you said, we kind of have to process it in a way that will polish it off and like get it ready for
Right.
translation, get it ready for movement out into the cytoplasm, and get it ready to be able to create a protein product. Um,
Yeah.
so there are a couple of kind of steps that go along with that, and I don't know. I'm trying to think if there's a specific order in which they happen, and I'm not sure
For the post-transcriptional,
for post-transcriptional processing. Do you know?
I know that it's not MCAT testable, so that's
Okay,
we
great.
can start there.
That's all we need to know as far as
Um,
concerned.
I'm trying to think, like, I would assume naturally the five prime cap would get added before the poly A tail just because the five prime cap gets exposed earlier. And that's just speculation. But yeah, there's three things that need to be done in
Mhm.
order for that mRNA to exit the nucleus. That five prime 7 methyl guanosine cap needs to get added. The
Which is an
poly
upside down nucleotide, right?
yeah, I don't know. I don't remember.
I think
Probably,
it's just a
maybe.
guanine that's flipped on its head, but
Yeah.
I don't
It
know.
is a guanine.
It's probably not MCAT testable either, but a 5' guanine cap, I think, is all you really need to know for that.
Exactly. The 3' poly A tail. Basically that's increasing the shelf life of the mRNA.
Mhm.
And it protects it against degradation, at least, temporarily. And then the splicing needs to occur where, we are getting those introns out and retaining those exons. And so those are the major post-transcriptional modifications. Just a quick note on the splicing piece, you know, the human genome, and I'm trying to remember this as accurately as I can. I believe the human genome is something around 20,000 genes, if I remember correctly.
That sounds pretty good. I would buy that.
I'll have to double check this. You know what? Let me just double check right now. How many genes? I'm pretty sure around
I
20,000.
like it. I like it. I'm gonna agree with you.
Wow, yeah, that was good. 19,900. So
Hey,
nice.
pretty
Uh,
close.
yeah. Um, Now the human proteome, though, if I remember correctly, is like in the neighborhood of 100 million. Let's double check that as well.
That's beyond me.
Uh, no, 100,000. Sorry, 100,000.
Okay.
Um,
More than 20,000.
yeah, and so the question now becomes, why do we have more proteins than we do genes? And
Hmm.
it's because these mRNA strands can be what we call alternatively spliced. Basically, it's mixing and matching different exons together to create different protein products.
Yeah.
It's like, if you buy, a whole like suit set, right? Like you could pair, the vest with the pants, or you could do the jacket with the pants. You could do the jacket and the vest and the pants. You could
I love
do,
this analogy. I love it.
yeah. And
Yeah, you're
it's
absolutely right. Yeah.
speaking from recent experience, maybe. But it's just different ways of mixing and matching, right?
Yeah.
You have the same set of stuff. You're just using some of it, all of it, or different combinations of it, and that gives you a lot of different outfits, right? A lot of different protein products, and so the proteome, the protein bank, we'll say, is just so much bigger than the genome as a result of that. And the last thing I would add to this conversation on post-transcriptional modifications is that prokaryotes don't have. Like prokaryotes, they have, concurrent transcription and translation where, as soon as that mRNA emerges from their RNA polymerase, the ribosomes are right on and starting to translate.
So much so that you can be like transcribing and translating at the same like on the same strand.
Exactly. Yeah.
Okay. Right? Like you could have like just the first 10% of the RNA like proceeding out of the RNA polymerase, and the polymerase is still going, and then you have the ribosome like jumping on as well, and they both kind of like move on at the same time.
The way you describe that, I don't know why, but for whatever reason, I visualized curling, where like you've got like
That's so
the
Canadian. That is so Canadian.
maybe. But like I'm not gonna try to even like name these players, but like the people that are sweeping out in front,
Yeah.
right? And
Yes.
then they're just kind of paving the way, and then I want to call it a curler, but I'm assuming there's a name there, and so if
Uh,
we've got any curling fans out there, I apologize. Please forgive my ignorance, but the idea being like, as soon as you kind of pave the ice a little bit, you're kind of right over it, and it's such a quick turnaround between the transcription and the translation, and it's possible for that to happen because there's no nucleus, right?
yeah.
You don't have to exit anything to be accessible by the ribosomes, whereas obviously in the eukaryotic cells we do.
Yeah. Beautiful.
And
I think there's a lot more to talk about. We've alluded to some things. We'll talk about translation next time. And like we don't know everything about all of this. Like we kind of talked about the vastness of the genome and how we don't know everything about what's going on. Like I spent some time in a lab where we were looking at these long non-coding RNAs. So they were like, thousands and thousands of base pairs long, or I guess not pairs because it was just a single strand, right?
right,
Uh, and they were present in like different tumors. Like we were working on this one. There's this
okay,
one that's really well characterized. It's called SHLAP, and it's like It's associated with radio resistance in prostate cancer. And so like
okay,
they work on these like targeted therapies to like attack SHLAP more or less. And if you like decrease the level of SHLAP in a prostate cancer, then it will respond better to radiation. And it's like, why does this
right.
exist? Why do we have these like RNAs just floating around that aren't coding for proteins? You know what I mean? And I think it's like an area that we don't know everything about. And so we talk about like there's so much of the genome that doesn't get coded into protein, but like it's doing something. At least parts of it are, and parts of it I'm sure are like just spacers. But it's kind of an
Right.
interesting area. I don't know. I've always found it interesting, but that's of course not necessary for the MCAT.
Yeah, no, I mean it's always cool though to bring in those connections because like sometimes we get so tunnel vision focused on this exam that we lose sight of like, well, like why does this matter? Like what does this have to do with anything? And I think at least for me as a student, like seeing what I was studying applied to the real world just gave me a better sense of purpose.
Yeah,
And sometimes like just finding little things like that to motivate you in your studying can be really, really helpful. But no, that's a cool experience. I think, you know, for anyone who has the opportunity to just explore different areas of research and explore different, you know, you know, styles of medicine, different specialties of medicine, like do so. Because like at a bare minimum, like it's just gonna make you more competent and well-rounded as a professional. And I'm sure we all have seen a doctor or at least on TV that you ask them a question about something outside their specialty and they maybe draw a blank. And while that's not wrong or bad or anything necessarily. It's always helpful to be, informed and, be able to contribute to, a number of different dialogues. But
yeah. I think that we have a tendency in like as pre-meds and stuff to like think that every experience we have needs to be to the end of like padding our maybe not padding, but like enhancing our presentation to admissions committees and residency selection committees and things. Like we want everything to be quantifiable so that we can like prove that we did something. And there's not anything wrong with that per se, but I think I agree with you. I just am agreeing with you that like you can take things seriously that are not necessarily like going to go down on paper as something you did, and it
yeah,
is like it's helpful. And I love what you said, like it will help your development or well-roundedness as a professional. I don't remember what exactly you said, but like
something
that, I
like
think
that.
we underrate that as med students and as pre-meds, but
Yeah,
I think those are good experiences to have, to make the most of whatever it is that you're doing.
for
However,
sure.
however impressive or not impressive it is.
Yeah, and I mean, my thought would just be, don't worry about what other people are doing. Like, do the things that you think will inspire you to keep going along the path that you're on.
Yeah.
Uh, and so, yeah, and I mean, I think that's a lesson beyond just the MCAT and pre-med experience. Is like, don't worry about what other people are doing. Just take care of your own business, and everything will work itself out. I think that's a lesson that I'm going to learn pretty quickly here in med school. Is like different people will be doing different things, and I don't need to be
Yeah.
doing what everyone else is doing because like I can guarantee you I'm probably doing two things that not many other people are doing in that, playing a varsity sport still and then working part-time.
Yeah.
You know, maybe a handful of people are doing one or the other, but doing both is probably not going to be super common. And so while other people are maybe logging many more hours of, shadowing or whatnot than I am, I have, the things that I'm committed to. And obviously, like, I'll, take care of those other, aspects of my medical education. But like what I'm trying to emphasize that to people here is like, you don't need to be somebody else. Like stick true to yourself and, um, let that be what gets you in. Like don't get in by being somebody else, get in by being you.
Yeah.
Uh, and how that relates to genetics and the expression of genes is maybe beyond the scope of my
Yeah.
training.
We're getting outside of it here.
But moral of the story is that the central dogma is DNA to RNA to protein, as Eric mentioned. Transcription is the process of writing down that RNA from a DNA template, right? We talked about template versus coding strands, antisense versus sense strands, and then the different things that we have to do once we're done writing that code down to allow it to be translated. And that will be the subject of one of our next episodes. Anything you want to add to that there, Eric?
No, well put.
Awesome. Alrighty then. Well, Thank you all for tuning in to another episode of the Jack Westin MCAT podcast. We'll see you guys on the next week's episode. In the meantime, happy studying and all the best.